Hi viewers, welcome to the Biotech Path. Today we are going to discuss a concept called oncogene addiction. How oncogene dependence opens new paths for cancer treatment. This is what we are going to talk about. As you know, cancer is a multi-gene disorder, which means there are a lot of genes involved in cancer. For example, in human genome, there are about 20 to 30,000 genes, out of which about 300 to 700 genes are associated with cancer. So it's basically a multi-gene disorder. That is one of the challenges for treatment also. Sometimes what happens is particular tumors depend on a single oncogene for their growth. Just like a person gets addicted to alcohol or some other drugs or something like that, these cells are addicted to one oncogene. That is why it's called oncogene addiction. This vulnerability is what we call oncogene addiction. This is the basis for certain targeted therapies. Okay, as I told you, what is oncogene addiction? That means certain tumors depend on one activated oncogene. What is the use for therapy? If you block this gene, it causes tumors to collapse. The normal cells are not dependent on this phenomenon. Only cancer cells are addicted to that particular oncogene. For example, HER2 new receptor. This is a receptor that belongs to the family of epidermal growth factor receptors. There are 4 receptors in this family. This is the second receptor. This receptor is overexpressed in certain cancers such as breast cancers and ovarian cancers. So in other words, these subset of cancers are addicted to this oncogene. Now, if you inhibit this oncogene, the tumor shrinks. That is what is shown here. So that is the basis for therapy. Now, why this kind of addiction occurs to cancer cells? There can be several reasons. It may be due to the oncogenic network being rewired on oncogene-induced stress, or it may be even due to loss of redundancy. As you know, one gene may be compensated by other genes, but that kind of mechanism, if it is lost, then the cancer cells become addicted to one oncogene. Similarly, I have given another presentation on synthetic lethality. You can look at it. When 2 genes are knocked down, the cancer cell collapses, not by knocking down one gene at a time. So if some such synthetic lethal pair, one of them is knocked down by mutation or something, then the cells become addicted to the other gene. So in that manner also this addiction can happen. So these are the various mechanisms by which cancer cells may get addicted to one oncogene. So what happens is signaling mechanisms are reorganized around that one oncogene. It acts as a central mediator of these growth signals. The alternative pathways become weak. This particular pathway becomes stronger. That we call as network rewiring. So as I told you before, the oncogene-induced stress causes metabolic and DNA damage stress. So survival depends on that particular signaling from that specific oncogene. As I mentioned earlier, the normal cells have multiple survival pathways, whereas cancer cells rely on this particular oncogene. Similarly, I mentioned about synthetic lethality link. So blocking the oncogene triggers a catastrophic failure and the cells start dying, maybe through apoptosis or other mechanisms. Some examples include HER2 new oncogene, I mentioned this, BCR-ABL, EGF receptor, epidermal growth factor receptor, ALK, it's a kinase, BRAF, another kinase. These are some examples of uh, addicted oncogenes. In the HER2 new addiction, which is very popular, it drives the breast and ovarian cancers, as I already told you. Targeting this gene with specific antibodies, which is commercially available now, is called trastuzumab. BCR-ABL oncogene is another example in chronic myelogenous leukemia, which is due to what is called a Philadelphia chromosome, and it is this particular cancer cells, it's a blood cancer, you can say like that. So this type of blood cancer, which is called CML, is entirely dependent on this BCR-ABL oncogene. Imatinib was the drug used. It was a big success story when the FDA approved it for the treatment of CML. Another example is this EGFR addiction in lung adenocarcinoma, sensitive to the inhibition by drugs. Similarly, ALK and ROS1 drivers in non-small cell lung carcinoma rearrangements, they also have show a strong dependence, and the treatment is through crizotinib. Similarly, BRAF, a mutation occurs where one amino acid is changed at the 600 position, valine becomes glutamic acid. This drives melanoma, so targetable by inhibiting this particular oncogene. Some inhibitors are available. So the therapeutic impact, as you can see, is what we call precision oncology. One problem is you need molecular testing. It's not just ordinary testing. You need molecular methods to test the presence of these oncogenes, and highly specific drugs have to be developed. The survival signals collapse faster if you give treatment like this. As I told you, cells can be killed due to apoptosis or other mechanisms. However, this therapeutic avenue also has certain limitations like drug resistance, the heterogeneity of cancer cells, and another drawback is not all cancers get addicted. Only certain, this is useful only in certain specific subset of cancers for treatment. The cancer cells become resistant due to several mechanisms. There can be secondary mutations, bypass pathways, phenotypic switching, and so on. So whatever we have discussed is kind of summarized in this presentation. As you can see, oncogene addiction means cancer cells rely on a single overactive oncogene to grow and survive. The classic examples are HER2 new in breast cancer, BCR-ABL in chronic myeloid leukemia, EGFR overexpression in lung cancer, BRAF in against melanoma and other cancers. And the challenges are drug resistance, and we need to perhaps look at combination therapy. But the advantages are it is having fewer side effects, and it becomes a personalized treatment. So the take home message is cancer may be chaotic, but sometimes it bets everything on one gene. Identifying and targeting that dependency remains one of the most powerful levers in what is called precision oncology.